species, but not all E. coli are the same, just like not all Canis familiarises are the same. And so, like the most infamous one is the 0157H7. And so this is the one you hear about in the news a lot. This is the one that causes illness. It's not the friendly one that we have in our GI tract. But if we have this particular strain of E. coli, well, let's, before we talk about that. Okay, the three people that have this kind of dog, where are you guys? There. There. Now, let's say you guys brought your dogs here to class, and then you left them up here in front, and then you guys left the room, and we mixed the dogs all up, and you came back here. Did you find your dog? Mm-hmm. Yeah, because they're not, even though this is the same breed, they look a lot alike, and maybe I can't tell the difference, but they're not genetically exactly the same. They're not clones of one another. And same thing with this E. coli 0157H7. Not all of these are going to be genetically exactly the same. And we'll get back to this here in a moment. In your reading in the Berkey's manual, you may have found that there was a type strain for a particular organism you were looking at. And that type strain is usually the first one that was identified that's in that category. And oftentimes it's the one that we know the most about, but it doesn't mean it's the most representative of all the things in that particular category. So the DNA fingerprinting, which is that full-field electrophoresis, can help us to compare DNA fingerprints, obviously, of different isolates of bacteria. So even after we have identified that we have Escherichia coli, we can tell if we have things that are the same versus different through that full-field electrophoresis. And this is going to come into play in some of the presentations that we have. It's also going to come into play a lot in our food microbiology unit, which is the following unit. And then, of course, the whole genome sequencing also can be used as well. Now, one problem with all of these things, like trying to figure out how related things are to one another and so forth by just looking at their genome when we're talking about bacteria, is that they've got that horizontal gene transfer. And so the genes they were born with aren't the ones that they have to go life. They get some new ones along the way. And so unlike our children are genetically related to both parents and aren't going to pick up some random DNA from somewhere else as they live their lives. And so it can be harder to really to track what is happening from generation to generation, because even within a generation there's change that occurs as time goes along. And so, and also, as we learned, bacteria can pick up genes not just from things that they're related to. They can pick up genes from organisms in different domains as well. So it can be really hard just to look at their genome and say how related are they to this or that, unless they're clones of that particular thing. Then you can say that they're clones of it, or that sort of thing, because there's this change that can occur so readily. And you may notice that in Berkey's manual, when they're grouping things, like you looked at your genus, they're giving you information that has to do with phenotype. What does the organism look like? What is it like? It's oxygen requirements, it's gram reaction, it's morphology, what it might be able to ferment, those kinds of things. So even though, you know, when we're looking at the domains, that has to do with evolutionary relationships, when we get down to these lower, more specific layers of classification, we're looking a lot at phenotypic kinds of things. So we've already learned these things here. but as you probably are realizing from what we're talking about, that we look at a lot of different things to decide where something falls into the whole scheme of classification. Phylogenetic has to do with evolutionary relationships, so the domain has to do with phylogeny, how related things are, evolutionarily speaking, they are to one another. And then from there, we look at a lot of phenotypic things, and sometimes some genetic things as well. Of course, the genotype of the organism plays a big role in the phenotype of the organism as well. And when we're dealing with prokaryotes, they are haploid, so it's not like they've got two sets of genes, you know, the two different alleles that are involved in getting to the phenotype. They've got the one gene, and they're going to get that phenotype based on that one gene. This is the book we have in the lab, and again, so this is going to describe all prokaryotic things that we have identified so far, which is probably just like a drop in the bucket of what's really out there. And then from there, it divides them into phenotypic groups, just like you guys saw when you looked at the Bergey's manual that's online. And so that Bergey's manual that's online is now what is used in place of the books, because the books, actually, they didn't get updated like that often. And today, you know, with the internet and everything, it just makes it a lot easier to update things more readily. And we're getting such a huge volume of information now. I can't even imagine how big the book would be now if we actually had a physical book for that. You can probably recognize that when you just looked up your organism by the alphabet, and then you saw there's like 500 entries there just in whatever the letter was in your thing. So we used to have the two Bergey's manuals, and now we've got our online one here. And that's a link to it, which you guys have already seen that. Now what about viruses? So I'm going back to the domains. This will play a role in our virology lecture later.